Okay, so in this first lecture, we heard from Kavita that there are three basic mechanisms of evolution. One, so reproduction, of course, heredity. There is natural selection, and there are mutations. In this lecture, I will focus on the first two facets, so reproduction and selection. I will try to introduce some of the mathematical methods that I use to study this mechanism. So I will start by just mentioning and not going very deep into it in the in vitro evolution experiments. In fact, in the last 30 years or so, and there has been a line of research which has been developed in which it was possible to do actual evolution experiments. Let's remember that adaptation experiments have been done by humankind for the last 4,000 years, if not more, since we domesticated plants or since we domesticated animals, and we selected them, we breeded them in order to satisfy our needs. This is an adaptation experiment of the kind that was mentioned also by Joshi in his lecture yesterday. Evolution experiments are different in the sense that you don't ask the life form to perform any specific task. You just ask to pursue evolution in a given environment which is not, say, in which not one particular trait is preferred to a different particular trait, but survival and reproduction is, say, successful reproduction is really the name of the game. And in this case, in the case of, in the last 30 years, it was developed in a number of labs, in particular in Lenski's lab. It developed a protocol in which it was possible to follow the evolution of several populations of microbes, of bacteria, over a very long time and see what happens. And so this is what I'm going to use every now and then as a sort of benchmark for the concepts that I'm introducing, that I'm discussing in this lecture. So let's see what is the uniqueness of the evolutionary history. So the idea is that there were a number of objections to the evolutionary theory put forward by Darwin, in the sense that people were saying, well, after all, evolution itself, the evolution of life, is a historical discipline, because evolution itself is not reproducible. So it's not like physics, in which we can reproduce experiments and then see that things happen, and then we can change the initial conditions and see that things happen in a slightly different way. And with these experiments, it is possible, in fact, to reproduce, to some extent, some evolutionary paths, and then check what were the different conditions that led to different outcomes. So in this sense, uniqueness, so this is Lenski-Travisano mentioned, uniqueness of evolutionary history is itself amenable to careful experimental analysis. And you have the luxury of making a prediction, and then you can test it. This is almost like physics. This is Travisano. So what is this evolution experiment by Lenski that I use just as a paradigm? Now there are many experiments, and this probably is the first generation. And this, say, sort of, the protocol is very heavy in labor, so probably the new generation of experiments will be much, much more powerful. So start from an ancestral population of bacteria, E. coli, which were selected from a lab strain. I don't remember how this lab strain was selected. And then out of that, they selected two subpopulations, one which can digest, say, arabidose, a kind of sugar, and the other one, this is a Ara+, and another one which does not digest this sugar. And the reason is that in this way you can, say, check against contamination in the transfer protocol that I'm going to mention after that. Then out of these subpopulation were taken 6 initial samples, one, and other 6 initial samples from the other. And so we had a total of 12 populations, 6 with ARA plus and 6 with ARA minus, independent, originated from a single clone at this point. And then these populations were subject to repeated transfer. So the idea is that every 24 hours, you inoculated a solution. This is not the ordinary lab medium in which you cultivate E. coli. It's a poor medium. In particular, it does not contain this arabidose. Arabidose is used only as a check. And you inoculate this medium with 0.1 milliliter of a solution containing the bacteria, and you let it grow. You let the bacteria grow for 24 hours, and then you transfer 0.1 milliliters of this solution into a new fresh medium. And you, of course, sample these population all the time. You can put them in a freezer to check them their genotype. You can sequence them. At the beginning, this experiment started in '87, and sequencing was very, very expensive at that time. So only few points in the sequence were sequenced. Now, say the frequency of sequence is much higher because sequence is now a cheap thing. And then you look simply what they decide to do. No, at the beginning, they were the bacteria were very unhappy because this is a poor medium. And the first thing they did was, in fact, to grow slightly in, so let's say, this is now old. I say this is 40,000 generation, but this is an old slide. And now I think there are about 60,000 generations, if I have computed correctly. And so they evolved in this glucose-poor environment, and there have been billions of simple mutations which arose. But of these billions of simple mutations, at this time, which was about 10 years ago, only 10 to 20 mutations have fixed in one or the other of the subpopulations. Some evolutionary trends are common to all strains. In fact, really at the beginning, it was observed that the main effect on all population was that the bacteria grew larger. And then they got rounder, and they had a higher yield, say, on glucose. And in fact, and the other thing is interesting that I think now there are 6 strains which have evolved into hypermutators, in the sense that the rate of mutation is much higher than in the wild type. I hope that Pozniakowski, who has worked on this hypermutation, will mention something more about that in the workshop. In particular, it's interesting to know that there was one major innovation that is the ability to metabolize citrate. It arose in one of on the population, in only one strain, around the generation 30,000 something. And this is a major transition. This is the ability to metabolize citrate, if I'm not wrong, is one of the diagnostic that distinguishes coli from salmonella. So this is really crossing a barrier. And we can be pretty sure that it was not due to some horizontal gene transfer from contamination, because essentially it was a sequence of what is the mutation that led to this major transition. It has nothing to do with any natural strain. To show this kind of adaptation, so you see these are samples in these beakers, and you see that this beaker is higher turbidity than the others because it has evolved to use the citrate which is present in the medium and that the other populations are not able to exploit. Yeah. they are all present here. They're all present here, moral, and there is one extra thing that Kavita mentioned, and, but did not discuss, and that is drift. And the fact is that you have a population which is reduced at each day. It is reduced to this 0.1 milliliter of, so you have a reduction in population size, which is called the bottleneck, and then there is expansion again. And this introduces a very strong element of drift, and drift allows to fix variance more easily than if you have a larger population. Generation is, say, it's a bit complicated. I will show you. You can count about 6 to 8 generations per transfer. And because most of the time is in fact spent in a stasis in which the bacteria do not reproduce anymore. I will show you. Huh? Stasis. So stasis means that, say, when the nutrients in the solution are eaten up, the bacteria do not reproduce anymore. So this is a stasis in this context. Fixation. Sorry, fixation. It means that you have a variant, okay, and if you have, for instance, for one gene, if you have more than one allele for the same gene, you say more than one variant of the same gene in a population, you say that the alleles are segregating. Then one of these alleles grows its frequency in the population. When it reaches a frequency 1, it's said that it is fixated. This is fixation. Is that okay? 12 independent populations.
So 12 independent populations are maintained. And then they presumably take out things from that and do experiments to find out what the properties of those populations are.
Yeah.
So 0.1 ml goes into the next one, and the remainder is available for doing experiments to see what it is.
Yeah, and it also, part of it is freezed as a historical record, so that whenever you find that some variant arises in the population which is evident, then you can go back to the ancestor and see what is the population dynamics of that variant.
I see. So every day they are taking out 12 samples and storing them as history?
Exactly.
So 12 into number of days is the number of stored historical samples, is that right?
Exactly.
I see.
And what do they measure? Eventually they will measure fitness. I will show you how they measure fitness. This is the, say, number one thing they do. Then they measure the things, and they sequence whenever they find that there is something interesting, and they sequence routinely now, I think, every week or so. So, but let's say now, let's go to these basic mechanisms. So this is reproduction. And in order to speak about reproduction, I just show you what is the growth. So the dependence of the population size on time for something like the bacterial population of the Lenski experiment. So this is schematic. You see that time is in hours, and this is up to 35, 36 hours. And here we have a log of the number of population, and we are looking at this 10 milliliters solution that in a beaker that we are considering. So when you inoculate the medium, there is, first of all, a couple of hours roughly of lag in which apparently nothing happens. So the idea is that you have the bacterium has to sense the environment and to adjust its inner machinery to be able to reproduce and grow. So this is a lag time, and then they start reproducing pretty regularly in a nice. So in this environment, they reproduce. The lifetime before reproduction is about 20 minutes, 20, 25 minutes, because it is a poor medium. And so this is called the exponential phase. And you see that in this exponential phase, what you have is it looks like a straight line on this log linear plot. And then they reach pretty suddenly. In fact, it's not, appears really, if you look at the diagram, it looks like pretty sudden stop. And this happens when the medium is exhausted. And you have so a very long stationary phase. If you wait long enough, then they start dying, and they die more or less independently from one another. So it is exponential distribution of lifetime. And so this, again, it looks like a straight line. And in fact, you see that 24 hours, you are about here, and the exponential growth lasts about 3 hours or so. So it is 6 generations, 6 to 8 generations, more or less. And then most of the time is in the stationary phase. Now, to have an idea, generation time is about, so what I said is about 20 minutes. And you can expect, say, if you imagine that you have a very large sample, you neglect fluctuations, then you expect that and you assume that the cells are synchronized, that is, that they, like, I don't know, like a battalion, they reproduce exactly every 20 minutes, then the expected number of individuals at any time t will be given by the initial number of individuals times 2 to the power t divided by this reproduction time, and this is the integer part. But of course, the cells reproduce when they want, so this they are not synchronized. So in fact, what you have is something like that, in which the different generations overlap. So this is mainly, I would like to use this to introduce the idea of discrete generations, which is synchronized, and overlapping generations, which you have different, the different generations are not synchronized, and they're called overlapping. And also, we shall often switch between discrete time and continuous time description. And also, we have to, in discrete time, the equations that we have are iteration equations, so discrete, no? And in continuous time, we will have differential equations. So just to have but just make an back of the envelope calculation. You have 72 generations one day. If in one day we keep in the exponential phase. So if we were able to keep the exponential phase for that long, what would we have? From one individual in 2 days, we would have 2 to the 10 to the 43 individuals, and this is a mass of 2 to the 10 to the 28 kilograms, which is 3,000 times the mass of the Earth. So that means that, I mean, it's not possible. Yes, please.
As you told that, first the graph, why that stationary phase means the birth and the death are same. That is so large, first question. And the second question is that the equation n0 2 to the power t by tau. So that takes into account only of the growth phase. Yeah. Not of the stationary phase. So how to know from the equation when from the stationary phase will start? So if you can elaborate that.
Okay, I can elaborate. I will do that in a few minutes when I discuss the Malthus-Fairholz model. But the idea is that the total size of the population in this kind of setup is determined by the amount of nutrition available to the bacteria, which is, of course, not intrinsic to the bacterium. It depends on how large is the beaker, how much sugar there is, and that sort of stuff. And so in the kind of diagram that I showed, I just wanted to show that if you do nothing and you starve the poor bacteria, then they, after a certain time, which is about 30 hours or so, they start dying. They die at random, but they start dying. So there is a lag also for dying, let's put it this way, which is about 36 hours. Yes? Okay, this is a very good question. The point is that we are accustomed to study bacteria in the lab, in a good medium. And of course, bacteria in the lab are very special guys. I mean, if you studied, I don't know, the behavior of lions in a zoo, you don't expect that their behavior is really the one that makes them livable in the wild. So we really don't know. I mean, we know much less about what the bacteria do in the wild than what they do in the lab. And in fact, there is a suspicion that while we are accustomed to focus our attention on the exponential phase in the lab, because that is what is exciting. That is where they reproduce and they thrive and they do plenty of things. It is much more important for the bacteria in the wild, the stationary phase, because most of the time they don't find a good medium, so they just simply stay put, waiting for the good opportunity. We don't know. And there is some studies of this. And so the ecology of the E. coli is, and of course, the ecology of E. coli is not independent of where they say on the host. And there is plenty of study about that, but I really don't know the details. I'm following this because this is where the data are available and they're easy to reach. But it's a very interesting question. Yes, more or less the way in which a bacterium divides is by lengthening. It's a kind of cylinder. It lengthens, more or less keeping the same diameter, and then divided in the middle. So roughly with the same rate, yes. But you see, the morphology slightly change in the experiment because they become a little bit rounder. They're not exactly cylindrical. So they are, say, rounder than the wild type. And so I don't know exactly if this, of course, there are very nice quantitative studies on the way in which ordinary strains of E. coli reproduce, because you can use microfluidics to make them reproduce in a pipe, in a micropipe, and then you can measure very well how it works. And this is pretty much what. Yeah, sure. Yeah, but by and large, you say that this doubling time is the time a bacteria needs to increase its length by a factor of 2. But the growth is linear in time. The growth of the bacterium is not exponential itself in length. Yeah, the growth in more or less, yes. No, as far as I remember, no.
Recent experiments on other strains.
As far as okay, okay, we can talk about that. Okay. Okay, now, so I mentioned that there is this reproduction, and then there is also death. We have to take into account death. So the Galton-Watson process is a way of describing what happens. So what happens to the population as population size as a random variable if we take into account reproduction and death. So let's first state the Galton-Watson process in discrete generation. So the probability that a cell, let's define Pn as a probability that a cell has n offspring in the next generation. Of course, the usual case is that it has 2 offspring because it divides in 2. But if you look at, I don't know, yeast, for instance, this is not, maybe you may have a general case. And then you have a P0, which is a probability. And P2 is the probability of reproduction with, splitting into. Of course, in general, we have that it must be a conditional normalization so this conditional normalization, sum over n Pn equal to 1. Of course, P1 also could be there, but. Now, let's ask what is the probability P sub n of t that the population size equals n at time t, assuming that at the origin you had only one individual. So n 0 is equal to 1. This is a convenient quantity that we are trying to evaluate, given, of course, this collection of these small p n's. The one quantity observable is the ultimate extinction probability. So we ask, what is the probability that eventually the population dies off? In this case, we are considering only the fact that we are assuming that reproduction and death takes place independently for each bacterium. So we are not considering the effect of the depletion of the medium. This is important. So in this case, we are considering that there are independent reproduction death processes for each lineage, if you like. Okay, how do we solve the problem? The problem is evaluating this PM of T, let's call, where M is the size of population at generation T starting from one individual at time 0. Well, the idea is that you start with one individual times 0, then you have k individuals at time 1 with the probability P of k. With small p of k is the probability that this individual had in one generation k offspring. And then for each of them, you can repeat the question. What is the probability that they have, how we call it, mj descendants t minus 1 generations later? And the probability of having exactly m individuals at time t is given by the probability to have k individuals at generation 1, the probability that, say, individual 1 had M1 descendants t minus 1 generations later, individual 2 had the M2 descendants t minus 1 generations later, and so on, and that all these numbers sum up to n. And this is the condition that is expressed by this Kronecker delta here. And of course, these probabilities are independent. This is the hypothesis that I'm making. So the probability is that they have a mj, so the probability that the j's offspring had the mj descendants at t minus 1 generation is given by this pmj t minus 1. And you have to take the product because of the independence hypothesis. So we have a nice iteration equation which looks formidable, but we are going to solve it in one step. The delta imposes the condition. You see, these are MJs, is the number of descendants of each offspring. Each offspring J has MJ descendants at times t minus 1. And then you want to have that the total number of descendants of the K offsprings must be equal to n. Must be equal to m, yeah. This is an m, sure. Yes, this is an m, right. Okay, thanks. Okay, so how? Yeah, there's a mistake. So there's a misprint here. This one, this n should be an m. M. Okay, so how do we do that? We use a very useful trick, the generating function trick, of which EJ spoke, but it's in a slightly different notation, slightly different concept. So we define this function, gamma z of t, of this variable z, which is just the sum over n of z to the power n times this probability that we are trying to evaluate. And we define an analogous function for the distribution of offspring size. And then you can simply say, take the equation that I've written before, and then multiply by z to the power m, sum over everything. So this is here, and you regroup your terms, and what you get is this very nice looking structure. You see that what you get is a sum over k, this is a size of offspring, probability of having that size of offspring, multiplied by this quantity, that is again the generating function for generation t minus 1. It's exactly the same expression, raised to the power k. So that if you look at this expression and you look at this expression, you find that this thing is nothing but the g, this generating function for the number of offspring, evaluated on gamma t at time 1. Okay? And then you find that, so this becomes a functional relation. It tells you that the generating function as time t is given by this function g applied to the generation function of the same value of z evaluate at time t minus 1. So you can propagate the generating function over the generation by this function g. We started with only one individual. With one individual, the generating function here at time 0 is simply equal to z. And so what you have is that the generating function at time t is given by g applied t times to z. And this is a mapping. You start with z, and then you apply g, and then you apply g again, you apply g again, and then you see what happens. And now, what is the probability, the extinction probability? The extinction probability is the probability that this, so you start with 0, no, you want to evaluate the probability of being 0. And so you start with 0, and then you apply this t times, and you see what happens. All right? All right, so what happens? So here I'm plotting. So z, let's take a z between 0 and 1. And this is the function gz. And then we have, so because of the normalization, with z is equal to 1, gamma is equal to 1. Because it's just the sum over all the probabilities. So we know that there is a point here, z equals to 1, g is equal to 1, and gamma anyway is equal to 1, right? And now let's look, you have two possibilities. You can also prove that the g of z must be a convex function. So that is, that it has a concavity with towards, upward. Now, you have one possibility. You look at the derivative of g at around z equals to 1. If this is smaller than 1, the curve g of z lies above the line line z, say g equal to z. You see this bisector. And in this case, you have that the only intersection between g and this line is 1. Why am I interested in this intersection? Because if I find a point in which applying g to whatever gives the same value, then I know that I have something that is not going to change over generation. So I'm looking at fixed points of this application. And fixed point of this application is the intersection of this curve with the bisector. And we know that there is one intersection at 1. The problem is whether there is another one. In the case in which this derivative is smaller than 1, there is no other intersection. Then we know that the probability, then the only intersection is at 1. But that means that the only value that you can have iterating this application is equal to 1. That means the probability of extinction is equal to 1. Let's look at the different case in which the derivative is smaller than 1. In this case, the curve starts below the bisector, but it's convex. And then you can see that eventually must cross it somewhere. You can prove that the value for z equals 0 must be larger than 0. So there must be an intersection. Because it is down, just imagine that you start from whatever value, for instance, in this interval of z, and then you go, every time you apply the transformation, you get smaller and smaller values, and then eventually you can end up here. And if you start here, you find larger and larger values until you reach here. So this is a stable fixed point. For any value of z, which is different from 1, you end up at this fixed point. So this fixed point is the probability of extinction. You remember, the probability of extinction, you have to start from z equals 0. And then eventually you reach here. So this is called, this regime here is called the subcritical regime, which leads to certain extinction. This regime here is called the supercritical regime, and it leads to exponential explosion of the population. And this is, of course, as we have seen for the case of bacteria, this is unphysical because it will lead to too many bacteria. I mean, it's two paradoxes. So this is incomplete. And there is, of course, a critical regime in which the slope of the curve is exactly 1. And in this case, you can see that it leads to extinction with probability 1, but with the power law decrease the population on average. We can also describe the Galton process in continuous time, and this is going to be useful. So this is why I'm doing it. Yes, please. Well, the fact is that you don't start with a single cell. You start with a population. So even if it is, say, large, then the probability that the whole population goes to extinction is given by the probability that each of them goes to extinction. So this is a very small probability. Okay, so this is just for, because it's useful, but and it is easier to follow if you have done the discrete time. So the idea is that, again, we have n of t. Remember, t is continuous, but n is an integer. And the probability given individual is replaced by k individuals in a short interval of time dt is given by lambda times pk of dt. And I put this lambda, which is a sort of a time scale. And with the sum over k, pk, pi k equals to 1. So pi k is defined for k equals 0, or k equals 2, 3, 4, and so on. And there is a probability to stay put, which is 1 minus lambda dt. This is a slight twist with respect to what we had. And then you do the same thing, more or less. So what is the probability that, so the solution, you look at the events in a short initial interval of duration dt, and then you have a probability of having n individuals at time t plus dt is given by 1 minus lambda dt probability of having n individuals at time t, plus the probability that something happens, plus something over the dt square, and then something happens has a lambda dt, the probability something happens, times sum over k, probability, so in p pi k, which is probability that it is replaced by k offspring, or k, which is 0, non-zero offspring. Here we have the usual condition that the total number of offspring must be equal to n in this case, and then we have again the product of the probability for the different size of the descendants of each individual offspring. And now you define the generating function in the same ways. And there is only one difference, is that here when you look at the form of the change of the generating function in this way, you find that there is this extra term here. And so that means that you have to define the application is no more gz, but is gz minus z. And then the evolution equation is given by d gamma dt is equal to lambda times this time u applied to gamma z times t, where this u is given by this equation. And again, you have to look at a fixed point. A fixed point now is easy because it is the point in which this u function vanishes. And you also have to look at the stability of a fixed point. And the stability you have if this derivative at the fixed point is less than 0. Because if you are on the right of the fixed point and you go back, you go left. And if you're on the left of the fixed point, you go right. So, of course, you have a fixed point at 1, and the derivative at 1 is given by the expected change in the number of offsprings. So if the expected change number of offspring in a short time interval is less than 0, then your population goes extinct. If it is so, and then z star equal 1 is the only stable fixed point. If it is larger than 0, then there is the stable fixed point at z star less than 0, which is the probability of extinction. So it's just the same calculation, and you are invited to redo the calculation for yourself so that you check it. And again, you can solve it graphically in this way. And yes.
Minus z, the minus z is the u of z part, please.
Okay, so you see here, we have to look at this equation here and take on the dt part. So on the left, so we have to subtract gamma z of t. So this cancels this one, all right? And then what we're left is something which is proportional to dt. And you find that there is a lambda gamma dt, and then there is also this bunch of things. This bunch of things makes up gz. But here you have a minus lambda dt times gamma again. Now this makes up this z here. Now, this is also, so one point is very interesting. Imagine that you have a variant, you have a population of size n, and you want to ask, well, what is the probability that it survives? Of course, if it is in a subcritical state, and then the probability is 0, and we don't have to worry. But if it is in a supercritical state, there is still some probability that it goes extinct. And this is the question that Sanjay asked before. So what is the probability of survival, eventual survival of a population of size n? Assume that you have only pi 0 and pi 2, so there is only fission. And define that this s in this way is 1 minus pi 0 divided by 2. Of course, if s is larger than 0, it means that we are in a subcritical situation, so we don't worry. But then you can write an equation for the survival probability. You just imagine what is the probability that one individual either dies or reproduces, and then you ask, well, for each of them I can evaluate again the probability of survival, and the population will eventually survive if either of the two or both. I mean, in either of two cases survives or not. So what you have is this equation. This equation is you see, the survival probability with n individuals is given by 1 minus s times the survival probability that there is n minus 1 in individuals. That is, you take into account the fact that one individual can die, or the individuals can reproduce, and then you have a 1 divided 2 minus s. And this 2 minus s here is just for normalization. We have a boundary conditions. The boundary conditions are simply that if you start with 0 individuals, the probability is 1. If we start with in infinite number of individuals, the probability will be 1. And then you can solve this iteration equation. You get a very simple solution like that. That the probability is given by 1 minus s to the power n. And for s less than 0, it's obvious that the probability is 0, but for s larger than 0, and you start with only one individual, you have that s1 is equal to s. And this will come in a different guise, is in different forms later on. And in particular, find the minimal population size such that the survival probability exceeds some threshold. Yes. And this is, you know this already in a different form, but this is very sensitive. Now, this is also interesting. What is the probability, what is the minimal size such that we are pretty confident that the population will survive? So we put a threshold, say 1 minus gamma, where gamma is larger than 0 but can be whatever. And then you ask, what is the probability? So you know that the extinction probability for n equal 1 is equal to this z star that we evaluated. So the probability that all lineages get extinct is z star to the power n. And this, we like that the probability that all lineages get extinct must be larger than gamma, so that the survival probability is 1 minus gamma at least. So n must be larger than n star, which is given by this equation, the log gamma divided by log z star. And when, in particular, we can use this expression with s, and you find this very nice result. So n star is given by absolute value of log gamma divided by s, is inversely proportional to this s. And this will be useful. Now, as you have seen that there is a problem, because if we let the population grow indefinitely, it will be submerged by bacteria. So what happens is that, in fact, in a given environment, no more than a certain amount of individuals can survive. And so that means that what the technical term is called there is a carrying capacity of the environment. Okay, the way I like it is that S is really the something like a probability of how to say, of growing. Say it's something like a growth rate, in fact, in the population size. Yeah, minus 1 minus pi naught over pi 2. So it is what is something like a growth rate per generation, in fact. And so that tells you that in order to be sure that your population is large enough that so the number of individuals which make you pretty sure that the population is large enough that it will not get extinct is inversely proportional to this advantage. Of course, the larger this advantage, the smaller the population you need. Right, yeah. Yeah, yeah. It can be negative, but it's...
If S is very close to 0, the growth rate is small.
Yeah, then you need a very large population. Yeah.
Reasonably sure
that it will not get extinct. Yeah. Then you is sufficient to have a smaller population. That's the idea. Okay, so we have to introduce the fact that you cannot have a population, infinitely large population, but this of course means that the population itself, say the reproduction this process is not independent for each individual. So in this case, I'm not going to treat the problem stochastically. I'm going to assume that we have a sufficiently large population, and we just look at what happens if we take into account the fact that the environment cannot sustain more than a certain size of a population. And so we introduce what is called the carrying capacity, and we ask, and we put now in continuous time, and so n becomes also a continuous variable, so that we can use differential equations. And the idea is that we can, simplest model we can make is that the reproduction rate that is now called R is a linear function of n, which vanishes when n reaches this carrying capacity. So this model is called the Malthus-Verhulst equation. And you can solve this equation and you get this, this form, which is in disguise is something that Kavita has already shown. So you start with n0 individuals, and then eventually you find that as t goes to infinity, this term disappears and you get k individuals, and that's it. So what you have is this typical sigmoid form. And here I put a solution of the Malthus-Verhulst equation for the case of the Lenski experiment, with the numbers from the Lenski experiment. And you find more or less after 3 hours, you are at 4.5 to 10 to the 8, which is the number of bacteria that are there in the stationary phase. So this is just, and so the time R is taken from the division time and so on, N0, so all the numbers are from there. R is the growth rate, which is related to the doubling time by log 2. Yeah, this is a 20 minutes for the division time, so number I put. Okay, I have, well, half an hour for selection. So this is Darwin when he came back from the Beagle voyage. So I like it because usually you see, a big beard and all. So this is how he looked and was strong and combative. And this is a dramatic photo. This is really the time in which he was developing the theory, so around the 1846 or something like that. It was a very tense time for him, also because his daughter died at that time. So you find that he's very, again, very combative, but very stressed. And okay, you probably know this quotation, but I'd like to reproduce it in full because in particular, the last sentence is usually forgotten. And so, can we doubt, remembering that many more individuals are born that can possibly survive, that individuals having any advantage, however slight, over others, would have the best chance of surviving and of procreating their kind? On the other hand, we may feel sure that any variation in the least degree injurious will be rigidly destroyed. This preservation of favorable variations and the rejection of injurious variation I call natural selection. Variations neither useful nor injurious would not be affected by natural selection and would be left as fluctuating elements, as perhaps we see in the species called polymorphic. So also the neutral case were considered by Darwin. First commentary was hinted by Joshi. My reflection when I first made myself master of the central idea of the origin was how extremely stupid not to have thought of that. So let's define fitness. Let's consider two kinds of individuals. Joshi rightly pointed out that selection does not act on individuals, acts on traits. So, but for the time being, let's accept that we have two individuals with A and B with population, say, kinds of individuals with population, NA and NB at discrete generations. And define fitness, this is called the Wrightian fitness in common parlance, as the expected number of offspring of individual of type I. So WI, to remind you that it is Wrightian fitness, where I is A or B. Assume that all individuals reproduce independently. The offspring of individual has the same kind, is of the same kind as its parents, and so there is no sexual reproduction here. And the fluctuations are neglected, which actually means that we are considering an infinite population size. Then we can write down deterministic equation, because we can neglect fluctuations in the sampling. And then, so we know that the expected, so the number of offspring of kind, so the number of individuals of kind I at generation t plus 1 will be equal to the number of individuals at time I at generation t times this Wrightian fitness. And we are considering that this infinite population, anyway, it's already infinite, will expand indefinitely. So that simplifies the calculation. So let's look at the change in the composition of a population, that is in the frequency of the different kinds. So the fraction of individuals of type I will be given, is defined as the number of individuals of type I divided the total number of individuals, where I and J are I of B. And then we just plug in these definitions in the equation we had before. We evaluate what happens at generation t plus 1. We know that on the numerator we have WA times NA, and denominator we have WA times NA plus WB times NB. Then we can simplify using the definition of fraction and generation in time t. So we have this equation here, where appears the difference between the two fitnesses, and eventually what we get is this equation here, which tells you that the fraction of individuals of, say, type I at times t plus 1 is proportional to the fraction of individuals at time t times its fitness divided by the average fitness at the previous generation. Is that okay? So the definition here of the average fit is given here, definition of the average fitness. This is just a normalizing factor. If we set we define S by this in this way as a function of the ratio of the two fitnesses, this we have this expression, and it is easy to see that if S is positive, then the fraction X of individuals of type A will increase in the next generation. This is what also Kavita showed. There is one thing that we would like, that we can obtain from this, and let's look at what happens to the mean fitness itself. So let's imagine a more general setting. We have K different kinds, each with its own Wrightian fitness, and define the mean fitness in this way. It's just the sum over different fitnesses times the frequencies of the different kinds. And then this you can do also as an exercise. You can plug in the definition and evaluate the change in the average fitness. Yes? Why the average fitness is important? That's a good question again. The idea is that in some sense, if the fitness measures the kind of success that a life form has in its environment. So the idea behind this kind of calculations that if the average fitness is larger, that means that the population is more and more adapted. But there is plenty of discussion about that, because one of the things that we have learned, and we'll go back to it, is that the absolute value of fitness is really not very important as long as the population does not get extinct. So probably this is a sort of misleading quantity. So this is problematic. Yes, please. There's no choice here. It's just the fact that if a kind say, thrives in a given environment, its fitness will be larger. Will have more offsprings. How we can interpret what? Okay, this is in fact, it's problematic. We can measure in some way, we can measure fitnesses, and you can measure it in the wild, not of individuals, but of traits. But anyway, let's say that you can measure it. But it is not really so clear that higher fitness means, say, being better in any sense. The only thing here is a sort of philosophical, in the sense that you have a quantity that you can assume you can measure, and that appears to have a tendency to increase. Now, I will discuss that. It's really very problematic, but I will discuss that. So let's say the bottom line is this one. It's not only that the fitness increases, but also that the amount of which it increases is proportional to the variance of the fitness itself. So in this case, with discrete generation, is the variance of the fitness divided by the average fitness, and these are all positive quantities. So that is always a positive increment. When the variance goes to zero, no, it's not extinct. It means that you have all the individuals have the same fitness, and there is no space for improvement. Yeah, then it stays. We are not considering the possibility of mutation. There are no new variation introduced in this model at this moment. So this is a fundamental theorem. It's called the fundamental theorem of natural selection. I'm simplifying enormously what it is, but this is the gist of it. So we may consequently state a fundamental theorem of natural selection in the form: the rate of increase in fitness of any organism at any time is equal to its genetic variance in fitness at that time. Genetic variance is a different kind of, say, it's not exactly the variance I showed before, but for us it will be okay like that. Professor Eddington has recently remarked that the law that entropy always increases, the second law of thermodynamics, holds, I think, the supreme position among the laws of nature. It is not a little instructive that so similar a law should hold the supreme position among the biological sciences. And so we see in physics disorder growing inexorably in systems isolated from their surroundings, and in biology fitness increasing steadily in populations struggling for life. Ascent here and degradation there, almost too good to be true. And in fact, it's quite doubtful. And if you take the word of very well-known evolutionary biologists, my own view is that it cannot play an important role in biology. So it's problematic, let's say. And I'm probably now more of the idea of Maynard Smith about this theorem that I used to be. Yes. In fact, there are the curves, and I will discuss about the curves. I hope I will be able to discuss about the curves in my third lecture. So it appears to increase, whatever that is. I mean, yeah. But the point is that you are really looking, say, evolution is about the relative fitness within a population. So you might have a situation in which the overall fitness goes down, and yet evolution still goes on by natural selection. This is in continuous time. I probably leave you this as an exercise. Anyway, the slides will be posted. So the idea is that doing the same calculation in continuous time, I will just show you the last line. So here the fitness is defined in a different way. This is important, so we need it. You see that there is a different symbol, is F, because it is what I call Fisher fitness and what Kavita called Malthusian fitness. But I prefer Fisher because it looks like an F. And so you remember that F is Fisher fitness, and is defined in this way. So you you define the rate of increase of individuals of type K with respect to time. So the number of individuals of type K with respect to time obeys an exponential law like this one, and the coefficient is the Fisher fitness. Then you plug in the definition of, as usual, definition of the frequency, and you ask what is the derivative of the frequency of individuals of type K with respect to time, and you find that it obeys this kind of equation, which you have this first term is just this one, but you have to take off a term proportional to the average fitness in order to keep the normalization of the frequency. And then you plug in, again, the definition of the average fitness, and you find that the derivative of the average fitness with respect to time is given exactly by the variance of the fitness itself, which is the form that Fisher was referring to. Now, what happens if we take into account the Malthus-Ferlus equation? Because here I didn't really consider that. So in this case, we just assume, let's simplify, assume that the carrying capacity is the same for everybody. And then you ask, well, what is the equation for the number of individuals type K? Yeah, it's given by something like that, in which we have to take into account the fact that the rate decreases when the total population increases. So this N of T is the total population size. And then you find that this starts being interesting, because then the equation of the total population size looks pretty much like the Malthus-Ferlus equation that we had before, except that the growth rate becomes frequency dependent. It depends on the composition of the population. You just plug it in and you evaluate it. You just sum this equation, and you find that this term here, so again, sums up as an average fitness, and there you get this average feature, so you can pull the average fitness out. Now we have a separation of time scales. On the scale determined by the average fitness, which probably we hope does not change a great deal, because say all the types of individuals which are in the population will be more or less adapted. So this will not change that much. And so over this time scale, you reach a population which is of order K. And then over longer time scale, you sort out the differences in fitness. Yes. Well, no. In this case, you assume that the kinds of individuals are given, and they have a given fitness. All right? They don't change over time. So the adaptation is at a population level. Fitter variants grow in frequency, and less fit variants decrease in frequency. So the population as a whole adapts to the environment. So why this selection, so the sorting out the differences here is lower, because the time scale is given by the differences in the Fisher fitness, which are presumably much smaller than the absolute value. Now, how you measure fitness? So I can make it. So we have really what you have to take into account is that fitness is a relative notion which depends on the environment in which you're looking at. So in this case, what you do, you want to look at what is the fitness of a strain, of an evolved strain, compare it with the fitness of the original strain, or probably of one of its ancestors. So in the case of the Lenski experiment, what you do is that you take a sample of the strain of which you want to evaluate the fitness, and you take a sample of the ancestor strain, whether it's the original strain or another strain in which with which you want to compare. I will tell you just in a moment, there's a problem here, which is quite important. Now, you mix these samples, and then in a one-to-one ratio, and then you put this, you distributed a sample of this mix on agar, and then you see how much they grow. So what you look at is the density of colonies. And now here it is the way in which you can use these, you can use the susceptibility to arabidose in order to strain the different strains. So you compare one ARA plus with one ARA minus, and then you can use this sensitivity to strain one with respect to the other. So you can distinguish, or you only have to count colonies. And then the ratio of the number of colonies gives you a measure, so the logarithm of the ratio gives you a measure of the difference in Fisher fitnesses. There is a problem here which is quite important, and that is the fact that here you are essentially measuring the rate of growth in the exponential phase. But we have seen that most of the time the samples spend the time in stationary phase, and something can happen there. So there is a problem whether the fitness you measure in this way has something to do with the survival to the reproduction process. This is a problem that probably Nagolsky is going to talk about, who knows much more about this experiment than myself. Now, we have assumed so far that fitness is a number which is attached only to the kind of individuals that we are considering. There are a number of situations in which you can imagine that something else comes in. So one of the possibilities is that it depends on the composition of the population itself. Let's put it this way. Imagine that you have a population which has a predator, and the predator is accustomed to see its prey in some form, yellow wings, for instance. Then you have an advantage as long as the larger part of the population has yellow wings. Then a butterfly which has, I don't know, brown wings will have an advantage because it will not be recognized by the predator. But if all the population has brown wings, then this is no more an advantage. So the advantage can depend on the frequency of the trait in the population. So in this case, what you have is something that is called negative frequency-dependent selection. If a trait is frequent in the population, the fitness decreases. And in this case, what you have is that because the difference in fitness between the two different kinds of individuals in population decreases as you go on, you find that there is some sort of equilibrium composition of the population, which is left unchanged by selection itself. So in this case, we have some form of a stabilizing selection coexistence in the population, not in the trait. And of course, you have the other possibility. You have a frequency-dependent selection where fitness increases with frequency. Now, you have a fixed point, but this fixed point in the middle is unstable. And so what you have is that your population will be either, well, essentially, depending on where you start within this context, we have a deterministic equation. So you start with this given frequency, you end up either with 0 or with 1. So if you imagine now that you have plenty of subpopulations, so you take a life form, you divide it into independent subpopulations, and then you let them go. Some of them will be one side, some of them will be on the other side just by different initial conditions, and then they will end up having different composition at the end, different stable composition at the end. So in this case, what we have is a several different possible equilibria, and it's called a disruptive selection at the population level. Now, something that was mentioned by, you can have the deterministic study of the effect of selection, of frequency-dependent selection at the deterministic level has evolved into an industry which goes under the name of evolutionary games, theory or evolutionary theory of games. So the idea is that the fitness, or if the fitness of a given trait or behavior depends on what the other individuals in the population, on the traits or behavior of the other individuals of a population, then you can state the problems of selection in this context in the same terms as von Neumann and Morgenstern described games, which were originally thought to be in the economic field. And in this case, you find a certain number of paradoxes. And here I will only mention one, which was already mentioned by Matteo in the first day, that is the prisoner dilemma. Prisoner dilemma, you have individuals in a population which have different strategies. Typical, one strategy, say, is a cooperation, and one strategy is defection. In the biological context, you can imagine this typical situation is the one in which you have that in order to exploit a resource which is in the environment, the individuals need to produce some intermediate, some chemical, for instance. Producing this chemical is costly, but it allows to take advantage of the resource which is in the environment. And of course, it pays for them to produce this chemical if nobody else does it. But if somebody else is doing it, because producing the chemical is costly, then it pays not to produce and to exploit whatever is around. So this is it's not exactly in the form of a prisoner's dilemma, but it's more or less what happens. So what you imagine to do is that you have something like first of all, you define something, a payoff matrix that it depends. You imagine that if there is a binary interaction between individuals, then you have that if cooperator meets a cooperator, say gets a fitness 1, if a cooperator meets a defector, gets a fitness 1 minus S1. If a defector meets a cooperator, it gets an advantage which is larger than 1, a 1 plus x2. And if defector meets a defector, it gets a fitness 1 minus c. And if 1 minus c is larger than 1 minus s1, then you have this dilemma that Matteo has mentioned, the fact that it is always better to be a defector because whatever the other guy does, you are always at an advantage. And so that means that in this case, you can find that WC of x is less than WD of 1 minus x for any value of x, and so the stable fixed point is defector frequency is equal to 1. And of course, the problem is that here the average value, so the final value of the fitness is less than you would have if everybody were cooperating. And then there is a problem, and why is cooperation maintained in nature? And what I just wanted to mention that there were experiments, but I don't have the time to discuss them in viruses. In this case, what you have is that if virus is alone in a bacterium, then of course it has to produce whatever is needed for its own reproduction. But if you have many viruses infecting the one bacterium at the same time, then some of them develop a defector strategy. And this was done in a beautiful experiment. And beautiful experiment by, oh, now I don't remember anymore. Anyway, I find it. And then this is very nice, but I don't have the time to discuss it. This is a rock scissor paper, which was observed in nature on these snakes in California. No, lizard in California, sorry. And it's a kind of rock scissor papers. So the nice thing is that this is a competition among strategies, mate controlling strategies among the lizards, but these strategies are also associated with color of the throat of the lizards, so they can be observed in the nature. And what you see is that you can follow the evolution of the fraction of the different kinds in time, so 90, 91, 92, 93, whereas 94 somewhere here, 95, you see that you find this oscillating behavior, because like in a rock, scissor, paper game. And I think I will stop here.
When you discussed about selection, and first we took the frequency independent case, and we got a formula where the fitness of a phenotype was constant, WA.
Yeah.
Okay, then the total physics or the iteration was only described by the mean fitness of the population, WX.
Yeah.
And the second case, when we went to frequency dependent selection, then both WX means the mean fitness and WA, both were X dependent.
Yeah.
So in the two cases, are the physics totally different, or the convergence time is different only?
No, the behavior, as soon as you have frequency dependent selection, you can all say at this deterministic level, all hell can break loose. So in the case, for instance, of rock, scissor, paper, you have these oscillations and there is no fixed point. So the population just keeps on changing its composition all the time without going anywhere. So you can have anything. And people have speculated you can even have chaotic behavior. I'm very doubtful about that. Well, at this level, probably, say, pattern formation in... I wouldn't like to say pattern formation. You would like to have something like a morphogen, which is say doing reactions and diffusing, and so this is probably it's a different kind of problem really. So I wouldn't use these mathematics for pattern formation. Yes.
The thing is that the thing payoff matrix that you payoff matrix prisoner's dilemma that you showed, yeah. And while explaining the payoff elements, you use the term fitness. Payoff element. Element of the payoff matrix. I'm talking of the prisoner's dilemma.
Well, say one of the solutions of a prisoner's dilemma, with which I'm basically agree, but is the fact that a prisoner dilemma is if you have one shot, then there is no way out. The only way is to defect. I mean, if your car breaks down somewhere in some places in which you are very unlikely to come back, you go to the nearest mechanic, it's very likely he won't fix it properly. Because there is no advantage to him, okay? You will be content if you can reach some known place. So so the way out is considering the fact that you have usually interaction between individuals go on and on, either because, say, you are in a confined region, or because you simply get to know somebody. Say, of course, if you have your mechanics is just downstairs where you live, then he will be very stupid not to fix your car, because he knows that you will not be back to him. You see? So if it is iterated, you can get out. Of course, there is some technical problem. If the iteration is for a fixed number of times, then you can propagate back the fact that the defecting is the best solution, then you're back to square one. So you must iterate for a random number of times. And you find that there are strategies which allow collaboration, which get advantages. So this is way out. And so again, you can find, there is a nice book by Axelrod called The Emergence of Cooperativity or something like that.
Yeah, the last question, as you told, sometimes a chaotic behavior comes.
No, just say that somebody had claimed that there is chaotic behavior.
Yeah, so what is the meaning of that in evolutionary context? Means if it is approaching to fixed point, I can have the idea one species is going to be fixed after some time, after a long time. But what is the meaning of chaos? Okay, so
actually, say one of the first non-strictly mathematical application of chaos was by May in an ecological context, in which he sort of generalized the Lotka-Volterra. Lotka-Volterra is a model of two species, prey and predator, interacting, which is very special and sort of very fragile. He sort of looked at a slightly more general case and found that it had some, say, sensitive dependence on initial condition. The phrase was not coined at that time, but okay. And so there was chaotic. Now, the problem is that this is in the context of deterministic equations. Now, if you look at populations are not described by deterministic equations. Populations are, as large as they are, they're always made of discrete individuals, and so there is always a problem of sampling. There is always a stochastic aspect to that, and if you add the stochasticity to a chaotic equation, what you get is a nice and fair probabilistic equation which goes to some sort of steady state. Now, just imagine adding some noise to the Lorentz model. What you have is a blob, which is nice and steady and does not exhibit chaos in this sense. So from this point of view, I don't think that what people have claimed that to have observed chaos in some populations. I don't believe that they observe chaos. First of all, because to observe chaos, really, you would have to have an experiment with slightly different initial conditions and see that they exponentially separate in phase space. And this is, of course, an experiment you cannot do with natural populations. Yes?
If two organisms are in a particular environment, and two of them have the ability to produce a particular substance that makes them to survive in that environment, that the cost of the cost of production of that substance is costly, so that if one is producing it and one senses that the thing is already available, that it can make use of that particular thing. So I'm asking, can we view a co-infection in the context of population genetics and a prisoner's dilemma?
Yeah, say, okay, this is, again, there are plenty of cases in which there is something like a prisoner's dilemma. One, say, for instance, one case also, instead of the producer, non-producer dilemma, is the case of warners. You have a population of birds, and they go somewhere to feed. And you find, say, I don't remember which birds was the observation, but you find that some birds do not feed themselves, but they stay alert just in case a predator comes. Of course, this is damages their fitness in the sense that they don't eat at least that time. They eat less. But it's advantageous for the group. And now you can make some sort of evaluation that if the group is connected genetically, then it is more advantageous to have a number of individuals which do not reproduce. Of course, the extreme case is the one of social insects. Social insects has brought this logic to the extreme case. You have that only the queen and one or few males reproduce. There was a theory that if the queen reproduced actually with only one male, then the whole thing made sense, because what you have is that the males in insects are haploids. So and the queen is diploid. So that means that sisters of the queen are more related to the queen than to their own offspring. So it pays for them to help the queen having more offspring than mating themselves in terms of propagating their own genes. Of course, this is anthropomorphic thinking somehow. And also, once they found that the queen does not mate with only one male, but with several males, then the whole logic is sort of disrupted. So, but you see that there is some problem. So I for instance, played with a model in which you did not need to have genetic relations in a group for the advantage of cooperators to establish. You only needed to have a group which is in itself, say, maintained. And my last work was the fact that you don't even need that the group is maintained. You can do that, then mix everything up, and then start again, provided that there is an advantage to have cooperators in the group.
So it's very interesting, this way of thinking of the Malthus equation now with your generating functions, right? But it is not clear still how you could explain the growth curve of bacteria, the lag exponential in stationary, right? So those pies, those probabilities of having specific offsprings should vary, right? A long time, or there is something happening there.
Yeah, well, you can say, by and large, you can explain the bacterial growth curve by a Malthus-Farrell's equation, but not really the details. In the Malthus-Farrell equation, you find that you have an exponential growth, and then you have an exponential tail when it goes to the stationarity, which have the same time constant. Now, if you look at the bacterial growth, you find that the going to stationarity is very abrupt. It does not. The time scale is not this characteristic time scale of the growth. So it's much more abrupt. So I wouldn't. In fact, what happens is really that the bacteria stop growing, not stop dividing. At a certain point, they switch off something and then stop pretty suddenly. It's not like this exponential decay. So that's probably, again, the model does not apply in the details.